With the help of the WWF Canada Restoration Fund, the Hammond River Angling Association has completed our first year of cutting-edge technology, using shrubs to sequester carbon and restore Palmer Brook. Palmer Brook, a major tributary of Hammond River, hosts declining populations of various species at risk, such as Atlantic salmon, wood turtle, and American eel. As part of our research on carbon sequestration, our team looked to sample carbon within the soil before planting began. We selected 3 locations based on their ecological state to collect data. Spaced throughout the timeline of the project, each site was visited 4 times and 8 samples were retrieved per visit. Each site is a 49-meter squared quadrat divided into a 4x4 grid, or 16 squares. Because this is a relatively large area, within each of these squares a grid was placed and a random number generator was used again to indicate where on the grid the sample will be taken. The first soil sampling site mirrors the site where planting will take place, an area that is categorized as seriously modified and is our priority for restoring Palmer Brook. These samples will serve as a base standard for soil carbon without our intervention, providing us with quantitative data to compare our long-term results to. The second soil sampling site is in a location with immature shrubs and trees, an ecosystem that is in a stage that we believe is realistically achievable within the coming years. The third sampling site is in a location with an area that is already established by mature shrubs and trees. This is important because we hope our restoration efforts will lead to an environment like this. Using an AMS soil sampling kit, we were able to retrieve soil to be sent for lab analysis. First, a hole must be augured to the desired depth by attaching your auger to an extension and cross handle. Next, switch your auger and cross handle for a slide hammer and soil core sampler with the sampling case within it. Finally, we cap the samples and label them based on which site they came from and the depth at which they were sampled. While the samples are being processed in the lab, there are a few more ways we can learn about our soils using field methods. We can test the general pH trends within the soil. If we use vinegar and observe fizzy soil, the soil is alkaline. The opposite can be done using baking soda to check for acidic soil. To investigate the composition of our soil, we use a soil texture triangle. To do this, we take soil from each site and remove any debris, rocks, and organic matter with a sieve. All that is needed to be done now is to add the soils to jars, add water, and wait. After around 48 hours, the soil will have separated itself into sand, clay, and silt. Based on the percentages of each and a bit of math, we can use the texture triangle to determine the category of soil. By analyzing the carbon in the soil in these locations, we can observe the difference these plants make in soil carbon. We can use this data in comparison with our planted area to observe its progress in carbon sequestration.